Good afternoon. Like she said, Leslie Henderson. I'm here to talk to you about the Territorial Coral Reef Monitoring Program. Thanks to everyone who showed up. This is close to my heart, and I'm glad to see that everybody here is interested in the same topic. So I didn't know I was going to get such a great introduction, so I put this slide up there because I haven't met a lot of you. I'm originally from North Carolina, but I've been on St. Thomas for about 7 years. I came down after getting my bachelor's at Wilmington, North Carolina. I came down to the Virgin Islands. I got my master's at the University on St. Thomas. I did my thesis on some caging experiments on the reef, but I worked on a wide variety of projects, anything from ciguatera, some fish spawning stuff, water quality, sedimentation. When you're a graduate student at UVI, you work on everything because there's not that many of us. So after that, I worked for UVI for a while as a research analyst, and I worked on this project. I was supposed to be sort of running it from the UVI end underneath my boss. And when I was approached by the local government to move over into the management side, this happens to be one of the projects that I continue to manage. So it's kind of an interesting, I worked as the person collecting the data, I worked analyzing the data, and now I'm submitting the proposal that gets the funding. So what is TCRIMP? TCRIMP has been around since 2001. It's not the only monitoring that's gone on around here. There's been lots of monitoring historically. There's lots of other monitoring simultaneously, but this program specifically started in 2001. And I put up our vision, which is to promote critical information, to provide critical information on the status and threats to all Virgin Islands coral reef ecosystems in order to increase management effectiveness and improve basic and applied coral reef research. So that's kind of a mouthful. I think it's a great vision, but really what is TCRIMP is more about what we do. So what do we do? Each year, a team of divers goes out and we have 33 permanent sites. We haven't monitored all 33 since 2001. There have been a few that have been added over the years as the program has grown. There are quite a few that were started in 2001. And you have a dive team anywhere 4 people to maybe 9 people. And you go out and you collect data on the corals, the fish, the algae, sea urchins, temperature. It seems like each year we're doing more for the same amount of money. And so this is a map that just basically shows all the sites. You can see where we've got a couple on St. John, but most of St. John is done by the St. John National Park. We have a nice spread around St. Croix. We have quite a few around St. Thomas. So we kind of get, and it's a depth range of about 4 meters to 40 meters. 220 feet, however many meters it is. 60 meters. Thank you. So I wanted to put this slide in here just to show you where the money comes from, because sometimes that's interesting. This is funded by the United States of America. Department of Commerce gets their budget. They then give money to NOAA. NOAA then divides up money into their different programs. The NOAA Coral Reef Conservation Program then has an agreement with local government down here. And so every 2 years we have to submit our proposal, and since 2001 we've been submitting for monies to do this project. We then subcontract the project because we don't really have the capacity to do all this work, but we want it done. So we subcontract the university and they go out and they do the fun stuff. So what do we get for all that money? It's about $150,000 a year to do this project. Am I blocking? Oh, I'm sorry. Okay. So what do we get from that? We get this report, and this report is beautiful, and I love it because I helped design the new model. But this report will go through and it gives summaries on a lot of the data that we're going to see today. It does site-specific summaries. Every one of those 33 sites, it goes through each one. And it also gives research highlights in the beginning for things that we may have seen in the past year or two that are interesting, or that we think are interesting. You may not, but hopefully you do if you're here. It also provides a lot of funds for UVI staff and faculty and students. There's at least 2 techs I know that get a portion of their salary, so we're supporting local jobs, I guess you could say. In addition to that, many of the people who go out on the boats and do this research are volunteers, you could say. They are students that want to get out, they want to get the training, they want to learn how to do coral health surveys, how to do fish surveys, and so they get to go out on the funds that are provided for this boat time. There's also a rich database that's been created from this project. So there's a lot of projects that will use the data that we get and analyze to characterize the site for another project, or to pick a site, or to piggyback on and say, well, you're doing corals there, I want to look at the sponges, but I need the information that you've already gathered in order to kind of build on the project that I want to do. So it kind of helps by having this really rich database. The other product is the information for management decisions, and this is what was stated in our vision, basically, is that whenever we have a management decision that might affect a reef, it's great to have local data about the specific reefs that might be affected. It's also good to know what your resources are if you're going to manage them. So this is our way of gauging how the reefs are doing and how they might be changing over time. Along those same lines, if the public, anyone, outside researchers is interested, this data is there, it's available, and you can get it by going to this website. So this website is sort of the data portal for the TCRIMP program, and on it you have access to the raw data. So it's been quality controlled, and thanks to John Jossert for setting this up. It's been quality controlled, and they upload, once it's been looked through, they upload the raw data. So when you download this, it's going to be like an Excel spreadsheet with the actual fish counts in it. There's also the reports, so if you just want a copy of the report, you can go and get that. And there's any publications that come from this data set, which there's quite a few. So, how are the reefs doing? I get this question all the time from a wide variety of people. And I am at heart a coral biologist, so I love turtles, I love fish, I love everything about the reef, but when you ask me how the reef is doing, I think, how are the corals doing? Because they're the backbone. And so when we talk about how the reef is doing, a lot of scientists will talk about coral cover. And coral cover is basically the percentage of the reef surface that is living tissue. So if you look down at the reef, like bird's eye view, and you were to estimate what percentage of that is living coral cover, or living coral tissue, that's the coral cover. And so we can get this by lots of different ways. You can lay down a quadrat, you can take a photo, but what we do, and what's becoming more and more common, is you do a video transect. So you swim along and point a video camera straight down, and you get a video of like a piece of reef. And so at these 33 sites, we're doing this every year for the same pieces of reef. So that's really interesting that you can actually sometimes, I have favorite corals that will appear over and over again in the video. So once you have that video, you then chop it up into pictures, single pictures like this one. And you will there's a program that will throw random dots over it, and you just write down what's under that dot. And so that's a non-biased way. You're not saying, oh, I think this is 20%. You're actually getting a scientifically accurate estimate for what the percentage of coral is. So once again, I wanted to throw this back up. This is all the sites. And now we're going to look at the coral cover at all of those sites. And if you're in the back, it might be a little bit hard to see. I remember watching some presentations in here and not being able to read anything. But you'll see that they're grouped into 3 large bins. And before I go over this graph, I just want to make sure we're on the same page about what types of reefs we're grouping them in. So the first grouping we use is nearshore. And so that's going to be a nearshore shallow-ish reef. And they're usually inside bays. So there's Cane Bay there on the left. And there's Sprat Hole there. We've got 3 St. Thomas reefs on the bottom. But they're usually inside bays. And so they tend to be more susceptible to land-based sources of pollution. The next category is the offshore reefs, and those a lot of times are around offshore keys, and they'll be named after their key. We have Savannah, not sure which one that one is. I think this is Castle. So around St. Croix, since you don't have as many keys, we'll do it on the outside of your barrier reef, and that'll be the offshore because it's not as directly impacted as it would be if it was in a bay. We also have Buck Island here. There's a couple. So that's the second category. And you can see that they tend to be they're a little bit deeper, and you can see they have a little bit more structure, a little bit higher coral cover, probably just removal from a lot of those land-based sources of pollution. And the third grouping that UVI is getting really excited about lately are the mesophotic reefs. And these are, mesophotic means mid-light, and so it's down where there's a lot less light than the previous types of reefs that I've shown you. So I think the most shallow one they have is about 100 feet. Their deepest one is like 225. So a wide range, but they all sort of have similar characteristics that they're low light level. And so the coral morphology flattens out. So you can look at these photos, you can see everything's kind of flat. These are the same species in those other pictures. They're just flattening themselves out so they can catch as much light as they can possibly catch. So they're very different. And in St. Thomas, they tend to be really far removed from the main island. They're a lot of fun. So now we're looking back at that same graph that I showed before, and now I have my representative reef types over here in case you forget. But this top here is near shore, then we have offshore, and then the mesophotic ones are on the bottom. And you can see in 2005, how many of you were here in 2005? Do you remember the bleaching event? Okay, so you can see that very clearly in our data, that we had much higher coral cover, and then right here, bam, it all drops. And that's 2005 to 2006. And we lost 50% of our coral cover in the nearshore reefs. 50% was gone. In the mesophotic, they only lost around 28%. They were a little bit healthier to start with. They were a little bit less stressed. Who knows why they did better, but they did. And so now some of the, if you look, some of the highest coral cover that we have is in those mesophotic zones. So that's great to look at everything all at once, but sometimes it's informative to zoom in on a single site. And so that's why in the new report we have each site laid out, and we're going to go through Cane Bay because I thought that might be an interesting site for you guys. It's a very popular dive site. It's a very fun site. It's very interesting. And so in case you didn't know where Cane Bay is, I put the map on here. And I love how close Cane Bay is to the Cane Bay deep site. So in St. Thomas, our deep sites are kilometers away from our shallow sites. This is kind of interesting how these two sites are right next to each other and yet they're so different. So this is Cane Bay, so we're going to look at this one, and this one in particular, and the coral and everything there. So the first graph is an informative one. It's one of my favorite graphs. This is the Benthic community, and over time, since 2001, we have continuous data at Cane Bay, which is not always the case for all the sites. And just before we go into it, everyone knows what a coral is, but we're talking about hard coral here, living hard coral, not the soft corals, not the sponges. We collect data on those, but this graph, we're talking about the reef building corals. And then we have these three categories of other benthic community members that tend to fluctuate, tend to influence coral cover, and can be really informative for what's going on at the site. So this photo here is actually from my thesis, and it's a tile that I had out that was inside a cage, and it grew a bunch of stuff on it. And it just clearly illustrates all three of these benthic community types. So the cyanobacteria, which is in the orange, is this black, it looks black in the photo, it's usually tinged red or it can be brown, and it explodes. It grows really fast, and it tends to be associated with high nutrient environments, so it can just bloom. It'll grow on anything. You can see it growing on the sand. You can see it growing on sponges. You can see it growing, here it's growing on top of some macroalgae. So it'll bloom and then it'll be gone. And so a lot of times in the cover you'll see it spike and then go down and spike and then go down. The yellow one, the next one on the list, is the epilithic algae. Now epilithic algae sounds fancy, but it's basically, when you go down to a reef, there's never a spot where there's nothing there, right? There's always like something growing. And so if you look at a piece of dead coral, there'll be like a thin fuzzy film on it. And that's the epilithic algae. It's turf algae. It's just a very well-grazed surface. And so it tends to be associated with areas where you have a lot of herbivores that are grazing. And it's good because it keeps down the macroalgae. This epilithic algae, if left unchecked, can develop into macroalgae because a lot of times mixed in the epilithic algae are tiny little seedlings of the macroalgae. And the herbivores love to eat them. They love to eat the baby macroalgae, but once it gets too big, then they don't want to eat it anymore. It's kind of like spinach. I love baby spinach, but like a full spinach leaf is kind of gross. So, the fourth one is macroalgae, and that we just talked about that and how it will grow and then it's hard to be grazed down. And so the example of that is this fleshy stuff here. This is the epilithic algae. It's just this fuzzy little bits. And so the way these three interact can be telling for what the reef is experiencing at that time. And it's really interesting. Now we're going to look at the graph, and you can see that coral cover dropped after the bleaching event. And after that, you really start to see this shift between the macroalgae increasing and the epilithic algae decreasing. And I can't say for sure why that's happening, but that seems to be happening on a lot of reefs. We're getting more and more macroalgae, right? You're seeing less of the grazed areas. And I don't know if that's because there's less grazers, or perhaps it's because there's a lot more space for them to graze now because the living tissue is now gone, and so that is now dead coral, and epilithic algae grows there, and it comes to macroalgae. But it is interesting to see that trend. And someone asked me before everybody got in here, are they recovering? Are the corals recovering? Well, based on this graph, and a lot of the graphs look like this, and the one we saw before did, you see the drop, and then you don't really see an increase after that. We're seeing a leveling off. And unfortunately, there's a million reasons for that, but the corals are undergoing warming events much more frequently than just this. We had a warming event in 2010 that thankfully was sort of dissipated when we got a hurricane that came and dropped a bunch of rain and shaded everything. So we'll look at some bleaching next, and you'll see that. But basically, these corals are not getting, in between the stressor events, they're not getting enough time to recover. And on top of that, they have the chronic stressors that are always there, like land-based sources of pollution and overfishing of herbivores a lot of times. So now we're going to look at the bleaching. So this is for Cane Bay as well, along the same timeline. And there's two things that are plotted here. We've got the bleaching prevalence and the bleaching extent. So the prevalence is, if you looked at the reef, out of 100 corals, how many corals had bleaching? So that's how frequent did you see bleaching. But the extent is, for that coral that was bleached, how much of it was bleached? Because we know it doesn't always 100% bleach. It'll just be partially bleached. You get 10% bleach. You'll get like a colony that's only bleached on the top, but the sides are fine. And so that's the extent. If it was 100% bleached, then it would be 100% extent. So you can see here in 2005, about 85% of the colonies were 85% bleached, and those are averages. So we had a major bleaching event. You can also see in 2010 this warming event where there were a lot of colonies that were partially bleached. So you can see evidence of that warming event, that the corals were stressed. Even though they didn't bleach all the way and die, they were stressed. And so it's important to understand bleaching is not just a finite death sentence. If you see a coral that's bleached, it may recover. It's not actually dead. What you're seeing is the coral tissue has just gotten rid of its color, but the tissue is still there. And so you're basically looking at a skeleton. You can see the coral's bones. And while that might not be a death sentence, the coral doesn't feel very good, and it may cause problems down the road. For example, disease. Dr. Smith, who's the UVI professor who runs this program, had a paper that came out shortly after this bleaching event that really showed very strongly that in the 1 to 2 years after a bleaching event, corals are much more susceptible to disease. So just like you or I, if you're stressed, you're not getting enough sleep, you're not eating the right foods, you get more susceptible to a disease. When a coral is stressed, it bleaches. So that's evidence that it's stressed. And then it's more susceptible to getting disease. So not only are these corals dealing with their bleaching event, but then they have to recover from them, and they're dealing with these invading diseases along the way. So you can see the bleaching event here, and then all of a sudden there's a spike in disease. And again, after 2010, you see this spike in disease. And progressively, our years are getting warmer and warmer, and disease is getting more and more common. And these are just black band here on the left, and white plague on the right, if you want to see what disease looks like. One of the other things that we keep track of in T-crimp is the partial mortality. So this is a coral. Usually when you see a coral, you'll see part of it's alive, and then there's also a lot of dead skeleton around it, unfortunately. And you can track this because over time, that old mortality should be decreasing as the coral grows back. You should see more living tissue and less old dead. Unfortunately, we're not really seeing that. They're staying pretty high levels of old mortality. And recent mortality is increasing a little bit. That might actually be, in my opinion, is more the researchers themselves getting better at identifying it. But old mortality would be, so for in this photo, you can see this is all living tissue, and then this would be the old mortality down here at the bottom. And I had another example, I think, of a larger coral where you can see the dead mortality mixed in. So this coral, you would measure the whole thing, and then you would put the percentage that's dead tissue. So here's an example of recent mortality. This is a snail that was eating this poor coral that I saved. And then here's another example of recent mortality, boat grounding that we responded to. So there's all types of ways for coral tissue to be killed. And so we try to keep track of how much is long dead and how much is just been killed. So now you can look at all 4 of those graphs all together, and you can really start to see what's happening on the reef and see the story behind that reef. So when the bleaching was really high, then you see this drop in coral cover. You also see the subsequent increases in disease, and you see the increase in old mortality. So you can really see how they all fit together to tell a story. And so we have these profiles for every reef in our program. So it's kind of fun to like go in and notice little things that you never noticed before. And so again, I wanted to point out how close Cane Bay and Cane Bay Deep are. We also keep track of temperature at each site, and the temperature probes actually record, I think, every hour, Rick? Every hour. And so we have, for most sites, we have temperature profiles. So this is what a temperature profile looks like, and you can see temperature on the left, the month on the bottom, and then unfortunately we didn't have these out in 2005, so these are different years. These are 2006 through 2011. And then you just see how the temperature fluctuates. You get a little fluctuation with each day as it warms and cools. And then you can see in the summer months, it starts to warm up. And then we get these peaks of the warm temperatures right here in September. And this line, this horizontal line, is generally thought to be the line at which if the temperature goes above that, then the corals are going to start bleaching, depending on how long it stays above that. So you can see the difference in these 2 sites. Even though they're really close together, one is much deeper, and it stayed much cooler down there, which makes sense if anybody's ever been free diving. When you dive down, suddenly it's all really cold. But this doesn't necessarily mean that those deep reefs are protected from warming. So this is from Dr. Smith's paper that came out this year, and this graph just shows temperature on the left, depth across the bottom, and then we have these 2 colors. You have the monthly maximum mean in blue, which is the average temperature on the reef that it's experiencing. And then you have the bleaching threshold in the red. And so this is created using all the T-crimp data from all the years and looking at when those bleaching spikes happened and what temperature it was, because we have all that data. And what he found was that the deeper you go, the lower that threshold is. So just because a reef that's deeper stays cooler doesn't mean it won't bleach if it's what it's used to changes by too much. So a shallow reef that's used to 29.5 degrees will bleach when it's 30 degrees, but a deeper reef, 40-meter reef, might bleach when it's only 29 degrees. And so this is really important when talking about deep reef refuges, because a lot of people have been supposing that perhaps the deeper reefs, since they don't get as warm, they might be protected from global climate change. But what this is telling us is that it's just not that easy, and that really the corals are specifically adapted to where they are. And when what the waters they experience become too different from that, then they may be stressed. So that's why you have, I'm sure you guys have seen corals on the mangrove prop roots, and it gets really warm in there, and they don't even bleach because they're used to it. And so that's just what this graph is showing. So really, if we look back at this, these two lines really shouldn't be in the same place because that deeper reef has a much lower threshold. And when you move the threshold, you start to see, oh, well, maybe it is going to experience some bleaching even at a lower temperature. Okay, so let's talk about fish. Before I get into fish, I just wanted to put out some caveats that we do about an hour survey, 10 units of fish surveys every year. And so fish, as you know, they move. And any fish biologist will tell you that if you're surveying once a year at one time on one day, you're not getting like a good estimate of fish stocks. You can't make a general conclusion about the fishery based on that one sample. If you go at a specific time of day, the fish may be somewhere else. If you come after the full moon, there may be tons of fish there that have all aggregated. And even if the water quality is bad, sometimes the fish will leave. If the temperatures get too high, the fish will leave. So relying on that one day of fish data is not a good way to summarize about fish stocks. However, we still do it because it's important to relate the fish to the coral. So the coral and the fish are connected, and so we still need to know what types of fish are at each reef and perhaps how they are interacting. So to collect the fish data, we have a diver that swims a 25-meter transect, and anywhere on either side of it, you write down what fish you see and how big they are, and that's basically it. For commercially important species, you'll put the exact size, but in general, it's just a what was here, what did I see, and then you're able to collect all that data from multiple people and kind of get a summary. So this graph is probably very hard to see, but that's okay because it's very general and it's honestly not super informative. This is the nearshore sites. They're all across the bottom, and this blue line separates St. Croix on the left from St. Thomas and St. John on the right. And in general, you can see that it fluctuates quite a bit, depending on perhaps the environmental conditions when we were there. You can also see we've been doing it a lot longer here over in St. Croix. And these are on the right, these are a bunch of sites that were sort of added later in the program. So that's why it's not that there were no fish in those early years, it's just that they don't have any data. King's Corner is y'all's fishiest site, in case you were wondering. Where is King's Corner? It's in the map I can show you, but it's basically on the west end. Yeah, maybe I shouldn't tell you. Pretty sure there was a mooring there for a long time. Is the mooring? I don't know if the mooring's still there. I can point it out when the map comes up again. So this is offshore. We have a lot more offshore sites over on St. Thomas, St. John. We have a lot more offshore keys. You guys have that very stark shelf, so there's not a lot of offshore. But you can see that it fluctuates. I feel like if you really want to get into the fish, you're going to have to try not to group everything like this. You'd have to look more at a specific species or a specific site and try to tease apart what's happening. The mesophotic is a little bit more different, and you can actually make some statements about it. You can see we have huge fish biomass here in St. Thomas on the bank. And this is all in biomass, so each individual fish is not worth the same amount. So a big fish is worth more. And so this is all like if you were to weigh all the fish, basically. So one bluehead wrasse is not the same as a red hind. They're weighted. Because what you want is biomass of fish. You don't care if there's 1,000 tiny fish, right? You can't eat those. So you can see that on St. Thomas, St. John, our mesophotic reef, we have really high biomass. You can see these huge spikes. And these are usually because the surveyors went out and there was like a spawning aggregation. I remember one year, I think this one right here was a bunch of cubera were just about to spawn that week, and so they were everywhere. But what's important to note about these is that Grammonic and Hindbank are protected areas. And so perhaps this is a result of that protection, that there's more fish there because they're protected and you can't fish in there at least part of the year. Over here on St. Croix, this is Salt River. I know you guys probably can't read it. This is Langbank. This is a closer Lang Bank one, and this is Cane Bay. And you can see that especially Cane Bay and Salt River are very low in fish biomass, and those are very easily accessible to spear fishermen. They're very easily accessible, and there's not a whole lot of habitat. So the habitat is very smushed together. You know, you don't have this wide habitat that we have. So here's all three, and I feel like this is too small for you guys to even read, so let's just skip that slide. So here's the map. So King's Corner is right here, out by Sandy Point.
Oh, by Sandy Point. Okay,
Yeah.
south. And
Right
there's
there.
a near shore and a deep.
No, that one is just a single site. Yeah, it gets pretty deep on the back end of it. It gets pretty deep, but the site is in the shallow area. But it's a really fun site. There'll be lots of big pelagics that seem to swim in, come in here, and hang out. And so that's why that biomass is so high. And it's also very well grazed. At least every time I've been there, it seems to be really low macroalgae cover. But I put this back up here because I wanted to point out the difference in the mesophotic reefs here and the deep reefs you guys have, how close they are. Look how close these are to your mainland. It's gonna definitely influence your fish abundance. And look how much habitat. Look at all this habitat. All this colored area is shallow enough for fish to live and hang out on. You guys have a wall there, which is an amazing wall, but it just drops out. And there's the passage there. Fish are not swimming across there. They're not swimming and hanging out down in there. So it's kind of interesting to look at the available habitat to a fish. There's a lot more up in this area. However, it's pretty neat that on St. Croix, you guys sort of have your own habitat, and that's it. Your fish are staying here pretty much all the time. Our fish, if we're protecting them, say right here, they might go to Puerto Rico and get caught. They might go to the BVIs and get caught. But St. Croix is sort of its own little system, and that's kind of neat as a manager because the decisions you make and the protections you put, you're going to directly benefit from them. So that's kind of an interesting difference between the two areas. So now let's look back at Cane Bay, just looking at fish. And these types of graphs are there for each site. And you're going to have, I can't even read these, but these are all the most abundant fish species in biomass. And so the winner at Cane Bay, the most abundant fish, was your Creole wrasse. And you may not recognize them just as two. You may recognize them when they look like this because they're just like giant big groups of them. But that was the most abundant fish. And it was a very abundant fish across the gambit for the entire island. Even on St. Thomas, St. John, it's very abundant. But it makes a lot of sense that it was the most abundant one, Cane Bay, because it's a planktivore. And so it likes to eat little bits in the plankton. And Cane Bay has all that upwelling because it's a wall. It has drainage very close to it, so there's a lot of productivity there on that reef edge. And that's where those fish love to just hang out and nip off any tiny little plankton that are there.
So you guys are looking at the whole water column when you're surveying?
Yes, you look up all the way to the top. I mean, you don't look straight up, but you look, you know, there's not a lot up there, but you do look up there. You'll see barracuda that'll swim by or a cerro, and you do include them. But most of the action goes on the bottom. So, lionfish. Lionfish is a hot topic for the past couple years around here. You've probably all heard the story. They were introduced in Florida. They made their way up and around, and they showed up here in St. Croix at Frederickstead in 2009. We saw them in St. Thomas 2010, early 2010. And on our roving dives, each site we also do one roving dive, which is like, or we actually do three, but it's the most fun because you just swim around and check off fish species. It's more of like a diversity census. And we saw a couple in 2010, but they started showing up in our transects in 2011. And so here you'll see, this is again is number of lionfish. So this is not biomass. This is actual number of individuals. And the only sites that are included are the ones where we have seen a lionfish on a transect. So this isn't all the sites. There's a few that we haven't caught one yet. But you'll see that basically over time it started to increase and then it kind of leveled off, especially in the nearshore and offshore ones. But in the mesophotic, it has just skyrocketed. And we did a dive there not too long ago at the Hind Bank, and they were just everywhere, huge ones. And so I'm not exactly sure why, but it could be that all the efforts from all the local communities to catch, kill, eat the lionfish are really doing a lot of good in these shallow ecosystems where you guys can access. But the mesophotic reefs are much more harder to access. They're 100 plus feet, especially on St. Thomas. You have to go really far. Dive boats don't go out there recreationally. So maybe that's the main difference between these different trajectories. So keep up the good work. Keep killing them when you see them. It could also be that something's learning to eat them. Maybe I'm hopeful. There has been reports of some groupers eating them. Some areas. I don't think there's been a report here, but something will eat them eventually. I'm confident. So we have lots of other data besides what I presented today. We have data on currents, we have data on sea urchin abundance, we have data on sedimentation for a lot of these sites, we have data on reef accretion. We're going to be doing some ocean acidification measurement collection soon. We have a researcher who's looking into recruitment at these sites. So we usually exclude any coral that's under 3 centimeters just because we'll be there all day. But that's an important size group, and it's really important to find out what's recruiting to an area, not just what's surviving, but are there babies, are they growing? And so that's a data gap that we're hoping to fill. There's also some talk about the Nassau grouper recovery, that they've been seeing a lot of juvenile Nassau grouper on St. Thomas. I think Rick counted like 70 in Brewers Bay. Was it 70 Nassau grouper?
Yeah, maybe around 60.
60, just in one bay. So that's pretty exciting, and we're hoping to catch that in some of the next couple years of fish surveys. This only had up to 2015, so we didn't see, I think they're going out tomorrow to do the final site for 2016, so hopefully that'll be available by March. We have someone who's going to be modeling the larval dispersal, so looking at when corals do reproduce, where do those coral babies go? Because if we can identify areas that are sources of larvae, then maybe we can protect those, or maybe areas that larva tends to be attracted to once it's ready to settle, we can protect those areas more as, as a priority areas. There's also a lot of sponges down here. I'm learning a lot from the UVI researchers that we have some of the highest diversity of sponges in the Caribbean, in the world. So there's some people who are working on sponges lately. Their abundance, their distribution, their effects on coral cover and all that nonsense. So there's a lot of stuff that we can do with this data. And if you want copies of anything, there's the website. You can send me an email. You can call me. I just want to send a quick acknowledgement out to Tyler and Marilyn over at UVI and Elizabeth and Razi, who are the techs who do all the organizing and data analysis. Also, my director, Noah CRCP, for funding everything. And I do have a video. Is there internet here?
Yeah. Yes.
If we're hooked up to the internet, I can play a video that'll show you.
I don't think that machine
Okay,
is hooked
that's
up
all
to
right.
the-
It's not a big deal. I'll put it on there just in case. But you can Google Ginsberg's Fringe, and there's an interesting video that shows some of our researchers down at that deepest mesophotic reef, and you'll see some of the huge lettuce corals that are down there. It's kind of a cool video. And that's it. So if anybody has any questions. Yes.
I was curious to see how little temperature difference there was in the water at the depth of 60 to 70 meters from the surface. Does it drop a lot more as you go deeper?
Yes, it does. And it also depends on the time of year. Sometimes you'll get a very strong thermocline where you go down a certain amount and then all of a sudden it'll drop like instantly a couple degrees. And so it depends on what time of year. But these temperature loggers are stationary. They're not going up and down, so they're measuring the temperature that they're at all the time. And so the difference in the Cane Bay deep and shallow was pretty significant for how close together they were. So if you were to look at our deepest site versus one of the sites that were in like a really shallow bay, there would be much more of a difference. But those two were right next to each other, and yet there was still a difference, which is what's interesting. But the water does mix. And obviously, if you were to go into the bottom of the Anegada Passage, it's way, 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 way colder. So,
paling as bleaching?
We count paling as paling. We categorize it separate. So we actually have like 3 or 4 levels of bleaching. So if it's just lightly paling, you can put LP, lightly paling. And then there's pale, very pale, and then bleached. If we've categorized it as bleached, that means it's stark white.
The whole thing, or maybe just?
No, that just means that there is stark white on it, and then you'll write down how much is stark white. And then if there's also paling, that one photo I showed, there was a coral that had lots of different, you would write 10% bleached, 20% pale.
Okay.
Is there enough data been collected to know whether the Eastern Marine Park has had an effect, a positive effect on fish populations, or as you call it, fish biomass?
The answer to that is probably no. The Eastern Marine Park hasn't done any evaluation of itself. So from the map you can see there's only 3 sites that we have inside the Eastern Marine Park, and there's only, I think, how long has Eastern Marine Park been around?
10 years.
It's only been around 10 years, and in the life of a lot of fish, that's not long enough to really see an effect. Some of the quicker growing fish, you might be able to see one if you had been collecting really good data that whole time. But there's also the very important aspect to remember of enforcement. So just because we drew a line doesn't necessarily mean that it's going to have any kind of effect on the fish until that line becomes reality and that there's nobody fishing in the zones that they're not supposed to be fishing in. I'll also point out that most of the Eastern Marine Park is open to fishing. So there's very small no-take areas that are just around the barrier reef. And so a large part of that, when you look at the map and it's that huge area, a large part of that is open to fishing, correct? Would you guys like to say anything about that?
Not anymore.
I think the National Coral Monitoring Program is another coral monitoring program that they have down here, and they tend to get a higher density of sites because they do much quicker surveys and they don't go back to the same spot. And so we're hoping that through that monitoring we might be able to see something eventually. They come and do it every 2 years, and they probably have 20 spots within the Eastern Marine Park. And so maybe, after the park is maybe 20 years old and we have 10 years of data from NCRIMP, we might be able to say something. But the short answer to your question is no, there's not enough data to say whether or not the park is doing any good. But I hope it is.
Any commercial information on fish catch or the
Yes.
success of the fish over time?
Yes, so the department, or the Division of Fish and Wildlife, has all the commercial data, and they rely on fishermen reporting that themselves. So there's not a lot of verification or fisheries observers. They're trying to get some of those going, but there hasn't been for a long time. And I know that the data history is not very well organized. I don't think it was a priority for a while, but now they're realizing we really want that data and we want it to be more accurate. So there has been a push, I know, within Fish and Wildlife to make the forms easier to fill out for fishermen, to start requiring them to turn them in more than once a year, because we were getting fishermen turning in their whole catch for the year at the end of the year, and it's probably hard to remember what you caught 1 year later. So there is a push to get better data on that, but I don't have access to that data. I could put you in touch with somebody who could talk to you about that, though.
If the fish data seems to be kind of the weakest link, is there? But it's still there, and for someone who might not be super well versed in this, but is still interested in the data, is there any concern that it could be like misconstrued as representing something that it's not?
I don't think that's happened. And I think that if you were to talk to any of the folks who are involved in TCRIP, they would be very careful to not say, well, look, there's no fish here at this site, but that doesn't, it's anybody who would read the fine print would say, well, this isn't really a good estimate of fish stocks. I know that we have so much data over such a long time frame that we're starting to get an idea of larger trends. And so I know Sean Caddison is working on a paper right now that's comparing the two St. Croix to St. Thomas and St. John, because there are major differences in the types of fish we have, and there's major differences in the abundance of some of the large groupers. There's many more large groupers on St. Thomas, St. John, and we don't see many of those over here. So she's currently writing a paper looking at that using T-crimp data, but that's like a bigger picture comparing over many years to large pools. And so when you start combining the data like that, you do get a more robust comparison. But if you're looking site to site, it's just very hard because, you know, the fish don't cooperate sometimes. Yes.
They do the surveys the same time every year?
They try to. They've moved it. So when they started doing it, they used to do it in the summer. But after the bleaching event, they started trying to target later in September so that they could see if there was bleaching. And so it's sort of moved from, they used to do it in July, and now it's more October. They come out, they come to St. Croix and they do all the St. Croix sites at once in 1 week in October, and then after that they go back to St. Thomas, and over the next month they do the St. Thomas ones.